Okay. Hi, thank you very much. We're certainly delighted to be here. I also want to introduce Joe Barone, who's sitting in the front, if you have any questions about our company. Yeah, Juventas is about 8 years old now. We're developing novel non-viral gene therapies. We have a significant platform that we've been spending the years building. What we have is a injectable non-viral DMA plasmid that can be used for a number of different indications. This plasmid secretes stromal cell-derived factor 1, or SDF1 in short. The technology was licensed from the Cleveland Clinic. We're moving forward. We're focusing now in a phase 2B program, which I'll tell you more about, but it's for peripheral arterial disease, which is a significant unmet need. We have existing data, though, in other indications, including cardiac disease, wound healing, dermal scarring, and other diabetic complications. We're going to be initiating our phase 2B study this quarter, and hopefully sometime this month or next month. I won't tell you a lot about SDF1. I think that it's been well recognized in the literature for its mechanism, which is to recruit endogenous stem cells. It promotes new vessel blood growth. It inhibits cell death following acute injury, and it also prevents and remodels scar tissue. The promotion of the new blood vessel growth is one of the primary mechanisms for which we're doing our PAD study, and I'll get more into that in just a second. In addition to PAD, of course, as I mentioned, there's a lot of other end organ systems that we can go after, and we do have some preclinical data in a lot of these indications. To date, we've shown our JVS100 to be safe. It's been administered to over 180 patients to date with no unanticipated drug adverse effects. We have a very highly scalable manufacturing process, and unlike the stem cell therapies that you're hearing about, we don't have some of those same issues in terms of cost or complications that are associated with the cell therapies. What we're using is a non-viral therapy, so it's a non-immunogenic. It does allow for repeat administration. Our drug, after administered, will secrete SDF1 for about 3 weeks and then goes away. Here's our pipeline. As you can see, we've advanced already a couple indications into phase 2, and one about to enter into phase 2, although we are going to focus on the advanced peripheral arterial disease, again, starting this month or next month. So let me spend a little bit more time now on the peripheral arterial disease. Well, it's a significant unmet need. One of the things that's happened over the course of the last 10 years is that there's been a significant decrease in amputation rates because the vascular surgeons have been able to salvage limbs. Where there's a significant need, however, is in healing the ulcers that are associated on the feet and other complications from the microvasculature that can't be addressed with some of these devices. There's over 2 million patients that have advanced peripheral arterial disease, and it contributes over $3 billion to annual costs. What we're going to be focusing on, though, is basically complementing the vascular surgeons in terms of what they do when they vascularize the lower extremities, and by providing closure rates for the wounds that are associated from the microvasculature. It's administered. It's a 10 to 15-minute procedure. It's a direct injection. It's at multiple sites in the lower extremity. It can be done as an outpatient. And it's a very simple procedure to conduct. The phase 2B program, which we're about to start, is built upon the phase 2A study, which we completed, which we called Stop Critical Limb Ischemia, or Stop CLI. This was focused on specific patients that were Rutherford class 4 and 5 patients who were poor revascularization candidates. We had 4 different dosing groups, and each of those dosing groups were placebo-controlled. This was an exploratory to try to identify the proper dose and use for our therapy. Our primary endpoint was safety, and we did meet that primary endpoint. The secondary endpoints were amputation, wound closure, and pain. Amputation rates, because they've dropped, was not something that was hard for us to evaluate because we had such a low amputation rate in our study. There were only 2 for the entire study. What's important, however, is that we showed trends in improvement at 12 months. And again, it's very interesting. It's a drug that only lasts for about 3 weeks, but yet we saw the improvements, which I think is important for therapy moving forward. The first slide here shows the decrease in the wound area, and as you can see, there's a trend toward improvement giving the 2 doses that we've selected, the 1 milligram, 8 injections, or the 1 milligram, 16 injections. Those were 2 of the that I had shown you. And also, there was reduction in pain 1 year after a single administration, and you can see that's in the second graph here, too. Again, comparing the placebo with the 2 different regimens. Our phase 2B trial, which we're about to start, is going to evaluate in patients with advanced peripheral arterial disease and non-healing wounds that have undergone a recent below-the-knee revascularization procedure. There are some examples and some pictures. I won't say too much, but we're hoping that we can, in addition to the revascularization procedure, then address the microvasculature so that we can actually heal these wounds. This is our trial design. It's a double-blind, placebo-controlled trial, 120 patients. We're going to use these 2 various dosing groups. They'll get 2 injections, one at the start and then again a second repeat injection at month 3. And we're going to be looking at them and doing follow-up at month 3, 6, and 12. The FDA has provided a fast-track designation for this trial. And we hope to complete the trial by the first half of 2017. So we're very excited about this trial. We also have potential, as I had mentioned earlier in the presentation, in other end organ systems, heart failure and dermal scarring in particular. So let me talk a little bit about the dermal scarring. Here we had, it was a phase 1B trial, which demonstrated reduced scarring and improved cosmesis at 6 months following injection. There were also 3 cohorts, also placebo-controlled for each of those. And you can see the trends that we saw in the POSAS scores, which are a patient-observed scar assessment scale for the patients at the various dosing groups. Also pretty impressive results also in terms of the wound healing that was seen 21 days after injection. Here's some pictures. I know a lot of times when we're talking about derm, people want to see the pictures of the skin, and you can see I think it's fairly impressive in terms of the placebo-controlled pretreatment and day 21 versus those who had received JVS 100, and I think it's fairly remarkable. For heart failure, we had completed a 93-patient trial. It was a placebo versus 2 different doses in patients with symptomatic heart failure. These were very sick patients. These advanced heart failure patients were on optimal medical management, which included all sorts of medical as well as non-medical therapy, such as ICDs and biventricular pacing. Also, very impressive results. It was safely administered to all 93 patients, no events, but there were trends in benefit observed for heart function, heart structure, and clinical status. As you can see on this graph, the results have been published. They were published just a few months ago. What was interesting is we looked at the sickest of those patients, those that actually had the lowest ejection fractions in that study. And what was actually very impressive was the magnitude of the changes that were seen relative to placebo. Cardiac function, structure, and the serum biomarkers all improved fairly substantially. And this was also been published, and as you can see, I think the results have been seen in the publications listed below. I'm going to end here with this slide in terms of what our partnering goals are. We're looking for a co-development and or commercialization partner for our PAD program. We're looking to bring in geographies that are outside the United States. We're also pursuing accelerated approval process in Japan and other geographies. In addition, we're looking for a development and commercialization partners for our dermal scarring and the heart failure programs. I want to thank you very much.